Hi, it's Dr. Robert Seichert. Welcome to a new episode of the Doctor Podcast Show. Thanks for tuning in. We really appreciate it. And we'd also appreciate it if you could click on the follow and the subscribe and the like button and the repost button as well, because the more you do that, the more interesting and great guests we get. Like today, we have our excellent guest, Dr. David Fajgenbaum. Dr. Fajgenbaum is a physician. He's the Associate Director of the Patient Impact Orphan Disease Center at the University of Pennsylvania, which is one of the leading medical centers in the USA and the world. He's also an Associate Professor of Medicine in Translational Medicine and Human Genetics at the University of Pennsylvania. He's also the founding director of the Center for Cytokine Storm Treatment and Laboratory, and we'll talk about what a cytokine storm is in a few minutes. And very importantly, he's the co-founder and president of the Castleman Disease Collaborative Network, and we'll talk some more about Castleman disease and Dr. Fajgenbaum's experiences with that. He's also the author of a national best-selling book called Chasing My Cure, A Doctor's Race to Turn Hope into Action, and we'll find out some more about that. Dr. Fajgenbaum has identified precision treatments for a disease and condition, specifically Castleman's disease, for which there was previously no cure or treatment. So David, thanks very much for joining us today. I know you're very busy doing a million different things. I appreciate the time that you took to come today. Thank you.
Well, thanks so much for having me. I'm really excited for this conversation.
Yeah. So tell us what inspired you to go into a career of medicine, and how did your experiences shape that and help you decide to do that? And tell us about your training, residencies, fellowships, advanced training that you had in medicine.
Sure. I had always been interested in health. From the time I was a child, I was really into sports. I dreamed of maybe one day playing college football. And
I
I
heard you
ended up,
were a quarterback, right?
I was, yeah, I was. I had the opportunity to go to Georgetown to play football there, and we're certainly not known for our football at Georgetown, but it was a really good place for me. But really everything changed for me shortly after I got to college. A couple weeks after I got to Georgetown, my mom was diagnosed with a glioblastoma brain tumor,
Oh,
and
sorry.
we. Yeah, thank you. We immediately knew that this was terminal, and obviously just heartbreaking for our family. So that for me completely changed the course of my life. I had thought about maybe a career in medicine, but really with the thought that it might be around sports medicine and health. And I sort of always had an interest in that side of things. But once my mom was diagnosed with cancer, I just said, this is what I want to do. I want to dedicate my life to taking care of cancer patients and also developing treatments for cancer patients. And so she ended up passing away about a year after her diagnosis, and I learned so much from her during her illness, and certainly before in her health. But that inspired me to want to go into medicine. And I after Georgetown, I went to Oxford and did a master's there, and then came back to the States, where I went to Penn for medical school. And I was 3 years into my medical degree at Penn when everything changed again. And that was not that a loved one was dealing with an illness, but I myself became really, really sick. So I spent many months in the ICU with a horrible disease called Castleman's, as you mentioned earlier. And that experience of getting so sick and then failing to respond to multiple therapies, which I'm sure we'll chat about more, led me to really understand that the only opportunity I had to survive if I wanted to live would be to get involved in research. And so I began conducting laboratory research and also clinical research. And I just fell in love with research. In fact, I fell in love with research so much that towards the end of my medical degree, I decided I wanted to go to business school at Wharton for a couple of reasons. One is so I could keep working in the lab for a couple more years while I was in business school, but also because I realized that some of the greatest barriers to research actually have nothing to do with medical technology or science. They actually have to do with the ability to bring together limited resources and get people to work together. So things you learn in business school. So anyway, I fell in love with it so much. I did that during business school, and then I actually got a faculty offer while I was in business school to join the faculty at Penn on the tenure track, which is
Wow.
typically more reserved for PhDs. But I had discovered a drug that saved my life and made some progress scientifically. So I sort of took a very atypical path to where basically I joined the faculty without doing a residency.
Wow, that's a pretty amazing story. So tell us when you got sick with Castleman. That was during medical school. What symptoms did you have, and how were you finally diagnosed with Castleman disease, which is a pretty rare condition?
Sure. So I was on my OBGYN rotation. And of course, anyone listening that's a physician, you'll remember OBGYN well. You get to deliver humans, babies into the world for the first time. And I did that and felt like I was sort of achieving a pinnacle moment in medical school. And right around the time of delivering the first child was when I started noticing I felt more tired than I'd ever felt before. I noticed some abdominal pain and some lumps and bumps in my neck that were lymph nodes. I also noticed these cherry hemangiomas popping up all over my body, like an eruption of blood moles, which of course blood moles are normal when they occur over the lifetime, but they're atypical to sort of erupt. So all this stuff was happening, and I was still trying to finish up my BUGON rotation, and the fatigue got so intense that um, I would go into a room to see a patient, and I would be so tired that as soon as I would see the patient, I'd need to find an empty room, and I would sleep in the hospital bed for like 7 minutes. I'd set my alarm on my phone for 7 minutes so
Wow.
I could sleep for like the full 7 minutes. I'd wake up, then I go to the next patient, then I go find an empty room and sleep for 7 minutes, go to the next patient, and I have to do that the whole day, and that's while consuming cups and cups of coffee and taking caffeine pills, just anything to try to stay awake. I just couldn't. So it was this incredible fatigue that I like, clinical grade fatigue. And of course, all of that sort of coalesced with me taking my shelf exam for OBGYN, taking the exam, and then walking down the hall to the emergency department where they did some blood work. And I'll never forget this ER doctor looking at me and saying, David, your liver, your kidneys, your bone marrow, your heart, and your lungs are all shutting down. We have to hospitalize you right away. It was like, how are you
Wow.
not hospitalized yet? So then I was rapidly transferred to the intensive care unit and had a retinal hemorrhage that made me temporarily blind in my left eye. I needed to be on
Mm.
dialysis 3 times a week, started getting daily blood transfusions to keep me alive. I got really sick really quick.
That's a pretty frightening story. How did they eventually diagnose Castleman disease? Because you had multiple
Great
organ
question.
failures.
Exactly. It's a great question. So I spent 11 weeks hospitalized before we got the diagnosis. And eventually the diagnosis was made because finally we did a lymph node biopsy. And, just as you said, I was in multi-organ failure. I was in the ICU. My ICU doctors were doing everything they could just to keep me alive, to stabilize me. And no one really knew what the exciting event was, what was driving this. And, some thought maybe this was a lymphoma. Some thought maybe it was an autoimmune disease. Maybe it was some infectious disease. And the more tests we did, the more things we were able to rule out, but we couldn't actually figure out what the heck it was. So
Right.
finally, a lymph node biopsy was done, and the lymph node biopsy came back that it was not lymphoma. It was a disease called idiopathic multicentric Castleman's, a hyperinflammatory disease where your immune system attacks your vital organs for an unknown cause. And it's called idiopathic because we don't know what ignites it in the first place, but it's just this incredible cytokine storm disorder. And now that we finally had a name for it, we could start treating it. So I started receiving chemotherapy, and chemotherapy finally got me into an initial remission, but then I relapsed shortly thereafter.
Wow. So tell us what a cytokine storm is. You mentioned that a couple of times.
Sure. Cytokine storm is the term that we use to describe when your immune system becomes hyperactivated and it starts to cause more damage than good. So you can think about SARS-CoV-2 as a great example where the vast majority of people that get infected with SARS-CoV-2 mount an appropriate immune response. They feel sick, they feel unwell, their lymph nodes might enlarge. But by and large, the immune system is able to control the virus, shut down the virus, and then get back into a homeostatic state. You return back to sort of the pre-viral normal. That's the vast majority of people. That is not a cytokine storm. You produced a lot of cytokines. That's what killed and helped to eliminate the virus, but that's not
Right.
a cytokine storm. A cytokine storm is with the exact same virus in someone else. In your attempt to mount an immune response to control the virus, you have this exuberant response where you produce so many cytokines, so many antibodies, so much cell directed killing that you actually cause more harm than good. Your immune response actually causes pathology. So then you end up in the ICU with multi-organ failure, your liver, your kidneys, your bone marrow, your heart, your lungs. It's basically a disproportionate and inappropriate response, which puts you into this multi-organ system problem. And so before the pandemic, there was a lot less attention on the concept of cytokine storms, because it turns out the vast majority of people who die from SARS-CoV-2 infection die from a cytokine storm, as opposed to dying from, like, let's say, an uncontrolled viral infection, for example.
Right.
So put a lot of attention on this. But actually, we started a center called the Center for Cytokine Storm Treatment and Laboratory about 10 years ago. We refer to ourselves as the Castle, and we've been studying this for about 10 years, and of course, many others have studied it even longer.
Right. So basically, your immune system goes totally haywire and starts attacking every organ in your body for some unknown reason. So it's like
That's right.
putting all the other autoimmune conditions all together, like arthritis
Yes.
and lupus and
Yes.
Sjogren's, and it's totally out of control. Well, it's good they diagnosed you, but they didn't have a specific treatment for it. Tell us how you finally figured out yourself, with some help, how to treat this condition.
Sure. So there was a randomized controlled trial that was going on of a drug called siltuximab. Siltuximab is an IL-6 blocker, and it's the first ever phase 2 randomized controlled trial ever done in idiopathic multicentric Castleman disease. Happened to be open right when I got sick back in 2010. I ended up not being eligible for the trial because I was too sick. I was on life support and was too sick to enroll. But I ended up getting the drug under compassionate use. So my doctor was able
Wow.
to get the drug, to me, even though it was off trial. And we were so hopeful this was going to work. Of course, I was very much out of it, but my family was very hopeful that it was going to work. And I got the first dose, and everyone sort of hoped that things would improve, and things just didn't. And I was getting so sick that my doctor ended up giving me a combination of 7 chemotherapies: adriamycin,
Got it.
cytoxin, etoposide, Velcade, dex, lutamide, rituxan. That combo of 7 to just obliterate my immune system, and that's actually what worked. I mean, I felt better with every dose of adriamycin, cytoxin, etoposide, which is sort of like crazy. If you're in oncology, you know that those drugs are some of the most toxic drugs around.
Right.
But I was so sick, and my immune system was creating such a toxic environment that actually killing my immune system with chemotherapy actually made me symptomatically feel better every day. My lab started to improve. It's really pretty incredible. But then I relapsed about a year later, and this was really important because I was on maintenance IL-6 blockade with siltuximab. So not only did IL-6 blockade not work in the acute setting, it now also did not work in the preventative maintenance setting, and which meant that the only drug in development wasn't working for me. And so I realized that if I wanted to survive, I would need to figure out a new treatment approach. And so I started a foundation called the Castleman Disease Collaborative Network to try to bring together researchers from around the world. And I also began conducting experiments on my own blood samples. So I had been storing blood samples leading
Really?
up to what ended up being my fifth flare of the disease every couple weeks. And those ended up really being what saved my life, because when I survived, thanks to a lot of chemotherapy, I could go back to the lab and look at those samples. I did serum proteomics, where we measured 1,000 analytes. I did flow cytometry, and did a cytokine panel. And basically, those data points led me to believe that the mTOR signaling pathway was turned into overdrive. mTOR, of course, is a critical mediator of the immune system. It helps the immune cells to communicate with one another, helps them to turn on, helps them to turn off when they should. And in my case, it was turned into overdrive. So we thought, well, maybe an mTOR inhibitor could treat this disease. It had never been used before for Castleman's, but maybe. And so I started testing an mTOR inhibitor called sirolimus, which you and your listeners are probably well familiar with. It's approved for organ transplant rejection and used off label for a few immune disorders. And
Right.
this January marked 10 years of remission on this drug that wasn't made for me.
Wow, that's an amazing story. So you did your own clinical trials on yourself, basically, and did all the laboratory research and everything else, and figured out a cure for your unique, extremely rare disease and condition. That's pretty incredible. Uh,
Thank you. I appreciate that.
Yeah, that's an
It's
amazing
been a
accomplishment.
journey, and you alluded to it earlier. It really was a team effort. I was a third year med student at the time, and
Right.
I had these amazing classmates of mine: Jason Ruth, Chris Nabel, Grant Mitchell, who were by my side. They were helping guide what the experiments would be, helping to interpret the data, helping to make decisions on what drug to try. I mean, it's pretty incredible.
Right. So is that drug now being used for other people who have Castleman's?
It is, yeah. We use it across patients all over the world. Unfortunately, it only works in about 20% of the people that we've given it
Hmm.
to. So it's not the it actually worked in the first three. So me plus the next two that we treated, it worked beautifully well. All three of us, very sick patients, all three of us very treatment refractory. Despite the fact it worked in all three of us, then we started seeing it in patients where it wasn't working. And so that's actually really driven the work that I do. So I continue to run the center at Penn, and
Right.
we focus in particular on Castleman's, but also another cytokine storm disorders.
Right.
Mm.
Now you mentioned you have a MBA degree. Did you get that before medical school or after?
I did it right afterwards. So it
Right
was
after.
when, yeah, so I did a master's at Oxford before med school, and then
What
I
was the
was,
master's in Oxford?
it was in public health, and it
Public
was
health.
yeah, it was public health with a focus on cancer prevention. It was pretty interesting. The UK in particular, but has sort of been ahead of us in the states in terms of thinking about ways to reduce the risk of cancer through risk factor reduction, so diet, physical activity. Of course, we've been leading in terms of smoking reduction, but the role the diet and physical inactivity can play in cancer is something that the UK really has led in. So went to Oxford to do that, and then it was the end of my medical degree that I decided to do my MBA. And as I
Wow.
mentioned earlier, it basically gave me time to keep working in the lab. So if I'd gone into a residency program, I was still battling this disease, and I relapsed in November of my first semester of business school. So that would have been November of my first year as a resident in my intern year, and so I wouldn't during my residency I wouldn't have been able to keep working in the lab. I mean, we all know that residency is so intense,
Right.
so I couldn't have been able to keep working in the lab, which going to business school allowed me to do. Um, but then I really think there were a lot of important lessons that I learned from my MBA that have really helped me a lot.
Right. So the basically the combination of your MD degree gave you the medical background, and then your MBA give you the business and entrepreneur
Yes.
background, and then combined with public health, all those three degrees have allowed you to now start doing research on other orphan diseases and other rare diseases. Tell us about what you're doing with that.
Sure. Yeah. So my center here, the Center for Cytokine Storm Treatment and Laboratory, we focus on a whole variety of inflammatory conditions, but all with that common thread of being hyperinflammatory. And then I also serve as the associate director of Penn's Orphan Disease Center, which looks across all rare diseases. So there are over 10,000 rare diseases, the vast majority of which don't have any treatment, the vast majority of which are completely neglected. No one's working on them. And so I've really leaned quite a bit into how do we create replicable models of advancing rare disease research. And so that might be, in one case, we crowdsource research ideas to determine, what's the best research idea from a community. Another is that we focus a lot of our attention on drug repurposing, for example. So it's thinking, how do you create models that other rare disease groups can just sort of pick up and take with them? And then more recently, about 2 years ago, launched another nonprofit organization, this one called Every Cure. Every Cure is a nonprofit on a mission to identify additional uses for existing medicines. So rather than just repurposing, for Castleman's, I knew the only way I could survive is I could repurpose a drug because I didn't have $1 billion in 10 years to create something from
Right.
scratch. Frankly, we didn't even have a target to develop a drug against either. So it's not like it was just I didn't have the resources at the time. It was like we didn't even know what to go after. But I knew that once we could figure out something to go after, my only hope of survival would be if there was something that already hits that thing, right? If that target could already be hit with an existing medicine, that's the only way I was going to survive. And this is done, occasionally. It's not done very systematically, but people like myself go on these journeys to try to find a drug. Researchers, physicians will say, "Hey, I think that Xolair, it works really well for allergic asthma, so maybe it could work for food allergy." And doctors will try things off label, and you get early, sense that that's the case. So this happens, but it's almost always based on serendipity. The doctor
Right.
happens to have this
You
patient.
stumble
The
on it.
patient happened, yeah, you stumble on it. There's no process for it. So we created Every Cure to create a process. So basically, we use the world's biomedical knowledge to rank and prioritize the probability of every combination of every drug against every disease being effective. So there's 3,000 drugs, there's 22,000 human diseases, meaning that there are 66 million possibilities for every drug to treat every disease. And so we start with 66 million. We use AI to come up with those scores. Now we've got 66 million scores. And now we can say, okay, among all those possibilities, which ones look most promising, and do it really systematically as opposed to haphazardly.
Right. That's an incredible story. Repurposing is really critical because there are many drugs, even in my field, ophthalmology,
Yeah.
where we
Yes.
find some drug or eye drop works for other conditions that other
Yes.
than what it was approved for. Another example is a drug called colchicine, which has been used for 100
Yes.
years to treat gout. And in the
Yep.
last few years, it's now approved for treating heart disease and preventing heart
Yep.
attacks and strokes in patients who are at risk for that. So there are many stories like that. Now, every cure, you mentioned, uses AI to figure out which drug would possibly work for which rare disease or
Yep.
orphan disease. Which AI system are you working with? Is it with one of there are about 5 or 6 famous ones now that are available. Are you working with one of the big technology companies, or you've created your own AI system for this purpose?
Yeah, great question. So we use a handful of different algorithms right now that I would say are the ones we use on a daily basis, or you would call maybe some of the quote unquote older AI algorithms. They're not the example of GPT-4, or I hear GPT-5 is coming out soon. They're some of the older algorithms and sort of the older approaches. So one of my favorite ones is a random forest model, and using a random forest model, which again is my favorite. So we start out with what's called a biomedical knowledge graph. So knowledge graph shows every drug, every gene, every protein, every disease, all in one two-dimensional space. And then there's an edge that connects every one of them. So it might be that IL-6 is associated with Castleman. So you've got an edge connecting IL-6 and Castleman. It might be that IL-6 is inhibited by siltuximab. So now you have an edge connecting those things. So now you have siltuximab inhibits IL-6, IL-6 associated with Castleman's. Now there's what we sometimes call a triplet, which is where now we've got three things connected
Right.
by a strong intermediary that could explain why now siltuximab might treat Castleman disease. So imagine that, but across all of biology, every drug, every disease, every gene. So literally tens of millions of edges between millions of nodes. It's really pretty amazing. So that's what we start with. And then we train the algorithm on the known treats relationships, meaning that we know insulin treats diabetes. So we train our algorithm to say, you know, that insulin to diabetes connection, there's actually many other connections between it. That's the pattern of a known treats relationship. We train it on about 9,000 known treats relationships. And then we unleash the algorithm. We say, okay, look across the rest of the knowledge graph and see if you can find patterns that are similar to the patterns where that drug treats that disease. And if there's no connections between them, give it a 0. You know, if there's no way to get from diabetes to a toenail fungus drug, that should
Right.
get a 0 because there's no path, there's no intermediaries, there's no connections. And so it looks for connections and patterns and it gives a score. And so, yeah, we're able to get a score from 0 to 1. And what the last thing I'll mention about that, which is pretty cool, is a random forest model. Exactly what we use is the same model that Netflix uses to suggest a potential next show that you might like. So, you know, if you're on
Wow.
Netflix, it's pretty amazing. You're watching one show, you go to the home screen and it says, like, you know, shows David may like, and you're like, man, how does it know that I like that show? And the way that it does that is it has come up with a profile for you. It knows the profile of the shows that you've watched in the past, the ones where you've watched all the way through versus the ones that you clicked off after 1 minute, and it understands the connections between you and those shows, and it says, you know, would there likely be a connection between your profile and this new show? And it applies it to every single show that it's got, and it says, this is the show, Robert, that you're going to want to watch. And it's pretty good.
Right. And it also might deliver some advertising
Yeah,
that you
exactly.
might enjoy as well. Speaking of Netflix, I think your life story and your accomplishments would be a good Netflix documentary or even movie.
Well, I appreciate that. We're actually
Yeah.
working on one right now.
Oh, there you
Yeah,
go.
so Wendy Feinerman, who produced Forrest Gump and Devil Wears Prada, and a number
Wow.
of other really, really great films, is working on making a film right now. So she's actually building out the team, and you never know what's going to happen with Hollywood, but apparently Wendy's working on it, and so who knows? Maybe a few years from now, there actually might be a show.
You look a lot like Tom Cruise. I think Tom Cruise could play you.
I appreciate that. You're gonna make me blush. As my number one concern is that it's not. I don't want it to be Andy Samberg because we look way too similar, and Andy Samberg is a very goofy comedian. And so, as long as it's not Andy Samberg, and I would love for it to be Tom Cruise. That sounds
Tom Cruise.
great. I don't think my wife would agree with you that there's any resemblance, but I appreciate that.
So speaking of AI, have you, with your MBA background and public health background, a company like Microsoft, which owns
Yeah.
ChatGPT, would probably be very interested in working with you. And I read recently that Microsoft and ChatGPT is working with Epic, which is the largest EMR system in the country. And I think, I don't know if you've already been contacted by them, but that might be a consideration because they seem to be very interested in the healthcare sector of AI, and maybe they could help you speed things along.
I totally agree with you. We feel so fortunate that Eric Horvitz, who's the chief scientific officer for Microsoft, he actually is an MD and a PhD, and he has been at Microsoft for many years now. He's on our board for Everycare, and getting Eric involved, I think, was such an important step for us as an organization because he is the one who's orchestrating the engagements with OpenAI and the engagements with Epic and all of these really cool R&D opportunities. And so we haven't formalized a specific partnership with Microsoft, the organization, but
Right,
we're thrilled to have on our board of directors Microsoft's chief scientific officer, and we think that's going to lead to some pretty exciting collaborations.
right. Now, why haven't the pharmaceutical companies done more research on repurposing their drugs that are already approved for disease A, let's say, and try to figure out what other conditions it might be used for? They certainly have the money and the background and the laboratories and the research capabilities and the personnel that's required to do that.
Yeah, it actually all comes down to money.
Money,
So when a drug gets FDA approval for one indication, the clock starts ticking on when that drug's going to become generic. And the moment that drug becomes generic, there now are going to be generic manufacturers of it. So the price of the drug is just going to rapidly fall off to cases where you might have 12 generic manufacturers of the same compound. And just based on economics, the price is going to just go down to pennies a pill, basically. And
right,
so if you have a situation where your drug brings you in pennies per pill, you have zero incentive whatsoever to spend what could be millions or tens of millions of dollars to prove that it works for, let's say, a rare disease that maybe has 1,000 people with it. So now you've gotten a
right,
thousand more pennies a year, and you've paid $10 million to get 1,000 more pennies a year. That's just the math just doesn't add up. And so,
right,
and it actually, in addition to that simple math, some more complicated math is that drugs get priced based on the disease that they're in. So it's that we do market-based pricing in medicine or in pharmaceutical industry. So when tocilizumab gets developed for rheumatoid arthritis, it doesn't matter how expensive it is to make tocilizumab, it's priced based on the rheumatoid arthritis market. So what's the cost of other drugs in rheumatoid arthritis? What's the current most expensive one? Is this better than that one in some way? Versus if tocilizumab gets developed for SARS-CoV-2 for the cytokine storm there, it's going to now be priced relative to drugs that are being utilized for SARS-CoV-2, which might be things like dexamethasone that cost pennies a day, right, or
right,
dollars a day. And so your price gets determined based on the market you go into, and you can't charge two different prices for the same drug.
right,
Once you get it into one market, that has to be your price for every other market. So by definition, if you say that you want to charge $40,000 a year for a drug in rheumatoid arthritis, you now have to charge $40,000 a year for, let's say, a drug that reduces acne. And the insurer is going to say, "Hey, I'm willing to pay 40,000 for RA because if you don't get it, you might be in the hospital. You have all these problems, but I'm not going to pay $40,000 a year for acne." So the drug company is now going to make a decision and say, like, "Hey, I'm going to go for this area versus that area." And so the math gets really complicated. And at the end of the day, companies are just doing the right thing for their investors, the smart thing as from a bottom line perspective. But it just so happens that in our current system, that means that we don't utilize drugs to help as many people as possible, which means patients like myself suffer while there's literally a treatment at their local pharmacy. So we're trying to
Right.
fix that here.
Yeah, kind of doesn't make sense. For example, I don't know if you've seen, there's some recent literature in the last few years that Viagra, which has been around for a
Yeah.
long time for erectile dysfunction, may actually reduce the risk of developing Alzheimer's. There are a few meta studies and statistical analyses that show that, but none of the pharmaceutical companies are interested in pursuing that because Viagra now costs less than $1 a pill, and
Yeah.
it's generic and they wouldn't make any money from it, yet it might prevent Alzheimer's. So that's a
It
big
it's
problem in
such a
our system.
it's a huge problem in our system, and it's such a great example. So Viagra is one of my favorite examples of repurposing. Many folks know that Viagra was initially developed for angina. It was developed to increase
Right,
blood flow to the heart, right? That was the initial indication, and also potentially as a hypertension medicine. Um, and then, of course, there was this unexpected side effect with erectile dysfunction, or erectile function was the unexpected side effect, and so that led to, a redirection as a company. But what most people aren't aware of is that Viagra was actually also repurposed for another use, and that's pulmonary arterial hypertension. So these are
right,
children that were dying as teenagers because of reduced blood flow to their lungs, and they were getting this horrible arterial hypertension. You give them low dose Viagra, and it completely opens up the blood vessels to their lungs and from their lungs, and they now live full lives. And that to me is one of my favorite examples of repurposing. Is that this drug that was developed for, or developed second line for erectile dysfunction, now is saving children's lives with this horrible lung disease. But then to your point, there's some really interesting clinical observational evidence that maybe it can reduce the risk of Alzheimer's. And
right.
like you said, the system just isn't built. The incentives
Yeah,
just aren't there to take a drug that you make $1 a day and then do a huge, potentially like tens of millions of dollars trial to prove it works in Alzheimer's.
right. But it's kind of interesting because the pulmonary hypertension drug, I think, is made by Pfizer. It's called Revatio, I believe, and that's a
Hmm.
branded drug. But you can
Yep.
get, it's a 20 milligram tablet of Viagra,
Yeah.
which you can buy generically for about $1. So I'm not sure how Pfizer makes money off of their branded product when it can be purchased generically. But I
No,
had an idea. What do you think of this idea
sure.
for repurposing drugs like that, where the pharmaceutical companies can't do it because they lose money? What about the NIH and CDC or FDA getting together and sponsoring a trial? They go to approach 10 medical centers, for example, like Penn, where you are, and
Yeah.
other major medical centers around the country, and they say, look, we're going to pay you to do this trial on patients
Yeah.
in your center to figure out if indeed Viagra does prevent or reduce the risk of Alzheimer's. Why can't the government sponsor those rather than having the pharmaceutical companies initiate that process?
I think it's a great question. I think the government can and should. I mean, you think about something, an entity like CMS that spends so much money every year on Alzheimer's. I mean, Medicare, huge amounts of money. I
Billions
don't
of
know, tens
dollars,
of billions, maybe even more than that, right? It's like
right?
some incredible amount of money. So if you say, okay, let's take 1% or 5% of what you spend every year, and let's do this study to try to prove, identify potentially a preventive mechanism. I think it makes a lot of sense. I think they're probably the only entity that can do that, because so one thing is that they're the only one who's going to benefit financially from doing this, because you're right, the drug company is not going to. However, as you know, prevention trials are the most expensive and the most difficult trials to do, because you need
Right.
to follow people for maybe 5 or 10 years, right? Whereas if it's a treatment trial, you can see, okay, I have Alzheimer's symptoms, did they improve? But a prevention trial is who's at high risk, and then did they get Alzheimer's? That's an expensive, long trial. And so the probably the easier short-term move would be to try to do a a relatively small trial of individuals who have early stage Alzheimer's, to see if you can randomize patients onto Viagra versus placebo, and see if you can maybe you could actually have some direct impact on active disease, which would then maybe help to justify this bigger trial in prevention.
Right. Yeah, I think something like that would definitely work and it wouldn't be as expensive. How long do you think your every cure system, the AI system, is going to take to find repurposed drugs? Can you guesstimate that, or it's just a matter of luck coming
Yeah,
across the right
we
one?
ran the first algorithm last March, and we got actionable results out of it last March. We couldn't believe literally the V1, the very first version. We've utilized a few different drugs based on predictions that came from that algorithm. In fact, a TNF inhibitor came up as the number one and the number three recommendation for Castleman disease, and we treated a patient with a TNF inhibitor last year, and it saved his life. He was getting ready to go into hospice care, and it saved his life. For
Really?
me, that
Wow,
was this incredible moment.
it's amazing.
Uh, really was. TNF, of course, is known to be important in so many immune disorders, and so maybe one could say, well, why didn't you think of that, David? Um,
Right,
but the bottom line is that there had not been a connection made for the 70 years. Castleman's was first described in 1954 by Benjamin Castleman. 70 years went by with no researchers saying, "Oh, I think we should target TNF." And this algorithm targeted it and said number one, and then amazingly, it came up with another TNF inhibitor, number three. They were like, wow, where did this come from? So we also had some parallel data from proteomics that led us and pointed us towards TNF, but that was newly emerged. It wasn't even part of the algorithm. So it bottom line is that this algorithm is able to produce predictions for drugs that can help patients. The question is, at what stage are we going to feel like we've refined it enough to where we're ready to sort of unleash it to the world? Because
right,
we recently got a federal grant from a group called ARPA-H to improve the algorithm and then to make the results publicly available. So within 18 to 24 months, we're going to be sharing every drug versus every disease, a score for how likely every drug is to treat every disease. And so, before we're ready to share it with the world, we want to feel really good about it.
right. Interesting. You mentioned ARPA-H. I did an interview with an ophthalmologist from there a couple of months ago where he's
Cool.
trying to do whole eye transplants, where you transplant an
Wow.
eyeball and then connect the optic nerve of the host, the patient, to the optic nerve of the donor eye, which has never been
Wow.
done. But ARPA-H
Amazing.
is funding that, and that would be incredible for many blind people who have no other choice or option.
Amazing.
Yeah.
Amazing.
Tell us about the book you wrote, Chasing My Cure, A Doctor's Race to Turn Hope into Action.
Sure. So I wrote it back in 2018. The New York Times ran a story about my journey in February of 2017, and that led to a number of book publishers reaching out and saying, "Hey, have you thought about writing a book?" And I said, "I've thought about it, but I've got too much other stuff to do. No time to write a
Right.
book. We got to focus on this work." But ended up getting a really great publisher, Penguin Random House, and a great partner and editor that I could work with, and was able to do it on my nights and weekends, mostly weekends. It was the year before my wife and I had our daughter Amelia, so I basically spent every weekend before our first child was born, working like 20 hours between the 2 days. So about 10 hours a day, every weekend for about a year, and ended up writing it. And my wife's been so supportive of me, and I'm so happy I wrote it. It's been able to help, get the word out about what we're doing, about repurposing, about so many perspectives that come from patients and in medical research that I'm so happy you've gotten out there.
So, Tom Cruise, if you're watching, please read that book because when you do
Exactly.
the movie, you'll need to know
You need to
all that
know.
background. So, this has been a very fascinating, interesting, educational discussion for me, and I'm sure the audience as well. We learned a lot, and we're looking forward to many future discoveries from you and your laboratory. It sounds like an incredible mission, and I'm sure you will be successful. Really want to thank you very much for taking the time today to speak with us. It's been great.
Well, thank you so much, Robert. I love the platform that you've built, and I'm so happy that I could be a part of this. And really enjoyed the conversation. I hope that anyone listening that's interested, go to everycure.org to learn more about our work, and go to chasingmycure.com to learn more about my work in particular. But yeah, just thrilled to be on this and appreciate the opportunity.
Right. I'll put those links on the show when
Amazing.
it's online so people can tune in. Can people donate money to your foundation?
Yeah, there's a couple ways that you can donate if you're listening. So you can donate financially at everycure.org. We're a nonprofit organization on this mission to identify new uses for existing medicine so people don't suffer when there's a treatment on their shelf at the pharmacy. But you could also donate data. And so right now, what that
Data,
means is that, yeah, right now it means that, like in your case, maybe you've received a drug off label, or maybe you've prescribed a drug off label to a patient and it worked really well. You can
right.
go to everycure.org/insights and you can share about that thing that you tried and that it helped you or it helped your patient. And that's a way just to share with the world. We're going to integrate those insights into our biomedical knowledge graph. And in the future, we'll actually enable the process for patients to share their medical data. So actual consenting to have parts of their medical record shared. But for now, it's just insights. So everycure.org/insights, you can donate funding and or you can donate your ideas.
That's a great idea, because if you get 5 or 10 people telling you they use drug A for their disease drug, for their disease B, you can actually figure that out. That's amazing.
I love it. Yep, that's exactly right.
All right. Well, thanks very much again. I appreciate it.
Thanks so much, Robert. This has been great.
All right. Take care. Bye.